Well, this is I was looking at the role of the environment in the transmission of the COVID-19. This is a very different topic. I feel like an outlier in this group. And I also only got the invitation last Thursday. So asked if I can be presented in this group. And so I throw together a few slides, and so maybe not as polished as some of the presentation you see in here, but I'm hoping to get a feedback and see a broader audience of how do you see this type of work, and guidance will be helpful too. Those are some of the research areas ongoing in my group, and actually I applied for this craft support twice, and first time I got some feedback, and it's on the quantitative microbial risk assessment. Actually, this is very much a framework and used a lot in the environmental domain, in the water industry, and it's all your water quality, water reuse, and it's being guided by recreational water, guided by quantitative microbial risk assessment, but maybe not so much heard in the medical field. But luckily, this proposal is funded by National Science Foundation, so I followed the suggestion from the committee and very much appreciate it to move on to a more sort of a technology-based approach and looking at the environmental transmission. So other things ongoing, and then towards this COVID-19 study in my group is also looking at, it's called sewage-based epidemiology. Maybe some of the words here are very strange to some of you, hopefully, and then we'll learn together and then through this little dialogue. And I also want to seek suggestions with this is called a multi-campus research project initiative, which is due this Wednesday, has a letter of intent, and maybe some of those shared with everybody. Feedback will be great because that will be reviewed by a very broad audience, very different disciplines involved in it. Well, I would, with the time limitation, I will first just introduce the work we have done with this QMRA, quantitative microbial risk assessment, since this is the most mature project I have been working on since February, way before all the fundings and outcries are coming out about COVID-19. The reason why we're doing that is in collaboration with Tsinghua University. This is the first come out in China for this type of, for this disease. And then some of my colleagues in Tsinghua actually reached me, and because I have certain reputations in the risk assessment and then throughout past decade in my career. We actually already completed a paper and submitted to the Environmental Science and Technology, looking at the risk assessment of indoor drainage system in a multi-unit apartment building as a transmission route of this COVID-19. And this is a newspaper article, and it's by CNN. It's maybe not newspaper article, but it's a news media, and talking about this concern, this outcry about this coronavirus spread through the bathroom. So they are putting on the investigation in this multi-story apartment building in Hong Kong. The outcry in the recent time is really, this is from February 12, 2020. It's mostly related to the early outbreak of SARS, the original SARS in 2003, because one case of a patient in one of the apartment building in this really large complex is called Emerald Garden, and then spread to 300 some patients in the 2003 case. And so now there's people in Hong Kong and then claim that they are getting sick because other people in the building get COVID. So this, in the 2003 investigation of the SARS is actually identified is the toilet flushing mechanism, because all our sewer system are connected. If you're in the multi apartment complex, maybe you go to apartment building, you may smell the sewer smell. Actually, the sewer smell is the pipe that's supposed to have a little water seal on the bottom. The seal is not there. So you can draw the aerosol from the sewage pipe, which is called the sewage stack. and into your apartment building. So this is the background of what those work. And again, if you just search coronaviruses and toilet flushing, you'll see a lot of newspaper articles report why you should flush with the lid down, and expert warn of fecal-oral transmission of COVID-19. And the reason why of this concern is actually more and more research papers show up from the medical field and indicating about nearly 50% of the patient actually shedding the coronaviruses, the SARS-CoV-2 in the feces. And then the live virus is actually isolated from the fecal sample very early on in China, since the February. And then there is a work have been looking at the load of the viruses in the feces. So there's quite a bit of work have been done in this area more and more recently. They also show the shedding in the feces is prolonged even after the swab from the nasal swab are gone negative. The patient recovered, there's no more symptoms. They still isolate viruses or isolate viral RNA from the fecal samples. So this is the base of our study. And then we're looking at the risk scenarios in the cases in the multi-unit apartment complex. The first scenario we're looking at, if you have a patient in a self-quarantine condition, you have a roommate or a suite mate sharing the same toilet. And then within there is a lot of work have been done about aerosol generation and toilet flushing. And so that's why you see those newspaper articles, and there's tons of them. And they did not do any real subjective risk assessment to it, but they point to that hint of why you should close the lid when you flush the toilet, or don't go to the toilet at all. So what we did in this graph you're seeing here, this is cumulative density function of, we collected all the reports. They have quantitative measurement of fecal shedding in human patient subjects. And then you fit that SARS-CoV-2 into a curve. This is a density function curve. And then we also collect the fecal shedding data from the original SARS from 2003, because there's all the data available through that. We want to compare the risk between SARS-CoV-2 and SARS-CoV original. And then using this data, basically this is a type of analysis that gave you a probability of the exposure based on when do you enter the toilet room after the first flushing. Say within 2 hours, there is a possibility of that aerosol to lasting in the toilet room. The second person enter that toilet room can breathe in the aerosol with potentially the COVID, the virus, the coronaviruses. And then, of course, this is based on a model analysis based on the aerosol generated from the toilet. I skipped a lot of those mathematics and then the aerosol generation and aerosol size distribution, but there are few direct measurements of the toilet room of aerosol for the COVID, for the coronavirus. And then this measurement from a toilet room in Wuhan hospital indicates there's 19 viral RNA per cubic meter of the air in the toilet room. So those, the backdrop, and then some of the data of the risk assessment that we're doing. This is a generic framework of the risk assessment. I did not replot this as a risk assessment framework with the COVID virus in here, but this generic framework include, of course, a lot of mathematics in there. Is basically hazard identification, identify what type of the biological property of those pathogen and hazard, and figure out the dose of that hazard in that environment. And then based on the human behavior, human activity, this can be either to touch the surface and touch your face, or inhalation. We have tons of study have done in the through human inhalation and the aerosol ingestions through a lot of other influenza and other aerosol transmitted diseases, and those are well-established area. And then based on those exposure assessment and then the dose response relationship of the certain pathogen, the dose of the pathogen causing diseases, you can derive of potential probability of illness. The probability of the illness, of course, is based on human model or animal model. It's impossible to have a human model to feed your viruses to a human. And then there isn't any animal model have been done for the current COVID-19 disease, but there was an animal model have been done for the original SARS. We can adapt that dose response model, but adjust what I have done in here to adjust for the affinity to the human cell, because both of those viruses using the same cell receptor, but their paper have been showing that the SARS-CoV-2 has 10 to 20 times more affinity to the ACE2 receptor. So adjust that dose response relationship by 10 times to use that relationship to arrive at the risk. So those are risk characterization. Basically, you have a single exposure risk. If you enter the toilet room, say within 2 hours of the first flash, and then, but only once. Or you can have multiple repeated exposure of the courses of diseases, say 15 days, and then you're always sharing the same bathroom, and then there's chances are increasing. That's called the risk characterization with repeated exposure. This is just explain how the basic model framework is set up. This is basically how the inhalation exposure is being proposed, is being modeled in here. So this basically a simple example of the dose of the viruses in the aerosol after flashing, and the volume of the flash water, and then the aerosol that human inhale based on the size of this aerosol, and then their the suspension of the aerosol in the distance where you sit on the toilet. That's the distance we modeled, and the duration, you either have a fecal movement or urination. So those are the duration of the time. That's just very fundamental. And
Sunny, you're about 2 minutes into the Q&A period, just so you know.
All right, I'm gonna hurry up. This is the dose response relationship. This is the outcome. So the outcome is the probability of illness. And then we compared this illness with the COVID-19, with the SARS. And then what you're seeing here is the median distribution of the results in here. I also plotted that special case, worst case scenario of where they directly measured the aerosol concentration in this. I just gone to the conclusion here. It shows the median risk of developing the COVID-19 during that two scenarios are generally pretty low. And they are in the range of 10 to the 7th, 10 to the 11th. And so that means the chances basically 1 in a million chances. But if you use the worst case scenario of the aerosol direct measurement, you have 1 in 1,000 chance of acquire the viruses and develop infection. So in all the models, you do a sensitivity analysis, the viral load in the feces are the most sensitive. So those are the paper that we have submitted in the review process. The second area, this is funded by the CRAFT, is looking at, it's called the sewage-based epidemiology. This is just being permitted for work. I want to understand if anybody heard of this word. Maybe not in this community, maybe in this group, but this sewage-based epidemiology is basically coined almost 10, 15 years ago. They used the sewage as an indication at the beginning is a community drug consumption for all those recreational drugs. You can see them in the sewage. You shed whatever you use. So those are basic sewage-based epidemiology. This is the idea to track the COVID-19 in the community. The reason why you can track the COVID-19 in the community is basically you shed it in your feces. You flush the toilet that go into the wastewater treatment plant. At the beginning of this work, I did it's called a backward calculation. Based on the number of cases reported in the city of New York, it's the epicenter in the US, the cases report. Based on the shedding rate in the feces, you calculate this rate, you feel like with the dilution factor in the sewage, you cannot see enough of the viruses without concentrate hundreds of liters of the sewage. But there are several preprints. I haven't seen a peer-reviewed journal come out with several preprints already reported the detection of the viruses in sewage. And what their authors suggest is they have a very high concentration of those viruses in sewage is because they are those non-reported cases, non-asymptomatic cases. And so with that, as we did set up this investigation, we have sampled from, well, we're starting the sample in the late April, and from 8 local wastewater treatment plants in Los Angeles, Orange County, and Riverside County. So that's our sewage-based investigation. We did sample collection. We did some viral DNA concentration. We did a cDNA conversion, but we're waiting for the pilot phase of this work to be complete in 5 weeks. And we'll send us 40 samples for deep sequencing, metagenome sequencing. What we're hoping to get is some map like this. You've probably seen this paper. This is a medical paper. They sampled, they surveyed 80 patients. They sequenced all that sequence, and they want to see where it's a source of the major New York cases coming from. From this sequence, they have identified most of those viruses from the patient in New York is linked to European origin. So this is what we hope to do with our sequence from the sewage water treatment plant, sewage treatment plant. So in the interest of time, I'm gonna wrap this up to say, we're doing, submitting on Wednesday, this letter of intent is focused on preventing the resurgence of the COVID-19 through modeling, surveillance, and preventive measure. In this effort, we're using a computer as a CFD model, and then to looking at the indoor air flows. And then we're using a bacteriophage called phage 6 and as an organism to see how it's dispersed in the room. And then that can trigger the cleaning action, either UV air cleaning or ozone air cleaning, and to see the efficiency. And then through that, we're also looking at sewage surveillance to indicate the resurgence of the disease and viruses in the community. So I'm gonna stop here.